So yeah, a few words about the last midterm. I have not finished grading them, so please allow me time to finish that. I hope to finish it by Friday. I have to go through everyone's exam and also do some statistical analysis still. Please give me till Friday and I will have those done. In general, what I noticed, people liked the 24-hour window to pick a time for the exam. Please make sure you take your exam during that time. There were a few people who had forgotten that the test was coming up, so with the final exam, please make sure you actually take your exam in that time period. So your final exam is Wednesday, May 6th. It opens at 9 a.m. and then it will close on Thursday, May 7th at 9 a.m. So what you'll see on Canvas is May 7th, 9 a.m. That again is the close date. So your exam is on that Wednesday. Pick a time to take it. What I'm going to do differently is I'm going to have the short answer and the multiple choice all within the same quiz document. So it will be two separate documents. It will all be in the same document. It will still be timed. You'll still see one question at a time in a random order. And the other thing I'm going to ask you to do is to download Proctorio and to take your exam on Google Chrome. So what this will do is it will lock down any other window browser so that you only can look at the exam window, and it's a free software, so it won't cost you anything to do that. What I hope to cut back on is the Google searching. When I started to grade the short answer, it became pretty obvious what questions people had Googled because you cannot just trust the Google machine. The answers that people gave that came from Google very obviously didn't even answer the question half the time. And they brought up material that I didn't cover. So you're supposed to be using the material that I have taught you in the form of the PowerPoints, especially in the previous homework assignments and the activities we've done. That is supposed to be the foundation that you're using to take your final exam. So again, it's chapters 1 through 8, it's cumulative. It'll be mostly multiple choice, some short answer in one exam window. You'll have 24 hours to pick your 2-hour exam window time, and you'll sit down and you'll complete the whole thing. Okay. If you run into a tech issue while you take your exam, the best thing you could do is try to screenshot what is happening and let me know, and I will work with you on that. Again, I'm not an IT professional, so I am limited sometimes in what I can do, but I will work with you if it's during business hours. If it's after business hours and you run into a tech issue, still screenshot what's happening, but I can do less. Okay. So all that being said about the structure of the test, I'm now going to switch and talk about the review topics. Here. So I got a fair number of questions from the review session, so this is a little bit long, but hopefully it is helpful. So the major question people want to know again was the action potential, the steps of an action potential. And so to start, we have to think about the neuron, and the neurons have an electrical potential. And what we mean by that is they have an ability to move ions in response to some sort of stimulus. And also, at rest, neurons are polarized. And this is not only true of neurons, as we talked about with skeletal muscle, and as we didn't talk about in A&P1, but you'll cover in A&P2, cardiac muscle also is polarized. And what that means is there is a distribution of charge across the membrane. And for neurons and for skeletal muscle, that means that there's more negative charge on the inside than there's positive charge on the outside. And this is driven in part by sodium. So sodium exists in a large concentration outside the cell compared to inside the cell at rest, and so that gives it that positive charge. And the negative charge on the inside is driven mainly by the stuff of the cell. So the DNA, the RNA, proteins, most of that is negatively charged. So the stuff of the cell makes the inside of the neuron negative. The sodium makes the outside of the neuron positive charge. And so this is not just true of neurons. Again, the same distribution exists in skeletal muscle and exists in cardiac muscle as well. And so for neurons, they're going to fire something called an action potential. And an action potential just means this wave of charge, this wave of ions moving. And again, other cell types fire action potentials. We talked about skeletal muscle, we didn't talk about, but it occurs also in cardiac muscle. What makes the action potential in neurons unique is its speed. So it moves really fast. We're talking milliseconds here. And its stimulus. So neurons can fire an action potential from a stimulus being light in your eyes, sound, those waves getting bent in your ears, pressure in your fingertips, or a chemical signal from a nearby neuron. Whereas other cell types, skeletal muscle, it only uses acetylcholine as its trigger, and cardiac muscle has its own pacemaker potential to it, which is a whole complicated story for AMP2. Okay. The stimulus, one, in a neuron, light, sound, pressure, a signal from a nearby neuron, will open two voltage-gated channels. And that's because the stimulus causes a little bit of positive charge to come into the neuron. That little bit, that change in voltage, is what opens these big voltage-gated channels, causing like a rush of ions. And so in step 2, we get sodium rushes into the cell fast, causing a depolarization, and then it inactivates. So it rushes in and inactivates because it is a little tired. And then the potassium channels will take over, and the voltage-gated potassium channels will allow potassium to flow out of the cell during a repolarization phase, bringing that positive charge back out of the cell. So sodium brings positive in where it shouldn't be. It's weird. It's depolarization. And the potassium brings it out in step 4. And this is now, if we zoom in on steps 2 and 3 here, we're looking at that voltage-gated sodium channel. So normally at rest, the inside of the cell is negative. However, when the inside of the cell becomes positive, whether because the action potential has moved down to where it lies in the axon, or because light has opened up some sort of channel, create a little positive charge right around that voltage-gated sodium channel, the voltage-gated sodium channel is going to open. Sodium is going to rush into our cell. But then it stops because that channel inactivates. This leads to our refractory period. So, excuse me, I ran my race, and I need a break now. And it stops. Okay. And if we look at it down the axon, it moves in one direction because of that inactivation. So sodium channels will open, sodium will come in, we will get a depolarization, and then it will inactivate, and so it cannot respond to that local depolarization. And behind it, we get a repolarization phase driven by voltage-gated potassium channels. So step 4, the repolarization is driven by voltage-gated potassium channels, bringing the ions charge back. So we now have positive on the outside again and negative on the inside. The issue is that the ion gradients are all messed up, right? And so we need to use energy, we need to use ATP to put sodium and potassium back where they belong in step 5. In step 6, we reach the axon terminal. This is the area where the neuron is going to have a synapse with something else, another neuron, a muscle, a gland, etc. And what happens at the synapse is that depolarization, that voltage change, opens voltage-gated calcium channels. And calcium comes into our neuron. And the calcium causes the release of vesicles full of neurotransmitter into the synaptic cleft. Okay, we're going to take neurons. That is neurons. We're going to put them on the shelf. We'll take them off the shelf in a second. Now let's look at muscles and talk about the structure of myofibrils. So a skeletal muscle cell is also called a skeletal muscle fiber, and it contains meaning muscle, fibrils, which are these elements of cytoskeleton, these repeating sarcomeres. We call the plasma membrane a sarcolemma. And the cell, if you look at it, is full of cytoskeleton. It's full of myofibrils. And they look striped. They look striated. They have dark bands that come from the thick myosin filament, and they've got light bands that come from the areas where there's just actin, just nice, thin, light actin. Okay, we zoom in again. Let's zoom in on that sarcomere. Sarcomeres run from Z disc to Z disc. In the middle, we have an H zone. In the middle of that is an M line. And when a muscle contracts, what happens is the filaments slide over each other, bringing the Z discs together. We're going to bring the Z discs closer together. So if we zoom in on what that looks like, we have our thick myosin filaments in the middle. These have head groups that will use ATP to generate a power stroke where they will move the actin to the middle. The actin is attached to our Z discs. There's nothing, when the muscle is at rest, there's nothing in the middle, no actin in the middle. But when muscle is contracted, that H zone will kind of disappear and the actin will come closer together. So how does this happen? Well, let's bring our nervous system down off the shelf, because what we're looking at is the interaction of a motor neuron on a muscle fiber, the interaction of that neuromuscular junction. So we know about motor neurons now. We know about action potentials in motor neurons, and we know that they can release a neurotransmitter. So if we start in the axon terminal, the voltage coming down, that depolarization, that sodium rushing in, will open those voltage-gated calcium channels. Calcium will come into our axon terminal and will trigger the release of vesicles of neurotransmitter into the synaptic cleft. At a neuromuscular junction, when we're talking about a motor neuron on a skeletal muscle, the neurotransmitter is always acetylcholine. So acetylcholine is the chemical released, and it's going to bind to an acetylcholine receptor on the plasma cell plasma membrane. And first, the trigger here is that acetylcholine is going to open a general cation channel. So acetylcholine binds to an acetylcholine receptor, which is a general cation channel. Anyone who's a cat can come on through, anything positively charged. It's dominated by sodium at first because sodium has that stronger gradient. It's got more of that driving force. But potassium can also move through that ion channel. And this will cause a change, a depolarization, a little bit of positive charge now on the inside of that cell membrane, the sarcolemma here in our muscle. And that will trigger an action potential in the skeletal muscle. And the action potential in skeletal muscle, while slower than the action potential of a neuron, follows roughly the same sort of formula using slightly different channels. So at the end, the action potential will stop when the acetylcholine is broken down by acetylcholinesterase, closing that acetylcholine receptor channel. And unlike neurons, which just send the signal in one direction, in muscle, the signal goes in both directions because it uses a different type of voltage-gated sodium channel. And so that action potential will move down the muscle fiber, But it's the same sort of action potential using voltage-gated channels. They're different channels than in a neuron. They're muscle-specific, but they're going to allow sodium to move in and potassium to go out, the same sort of depolarization, repolarization. Okay, so what does that do? Well, we have what we call excitation-contraction coupling. Excitation, the action potential, is linked to contraction of the muscle, and the muscle will have sliding filaments. So the action potential will cause the release of calcium from our sarcoplasmic reticulum, and that calcium will allow myosin to reach up, grab the actin, and pull it to the midline. At rest, the myosin can't do that. It's blocked. The actin is blocked. But calcium releases the block and allows the myosin to grab and pull on the actin. And again, the calcium is released from the sarcoplasmic reticulum with that depolarization, with that action potential. Myosin needs two things, though. It needs calcium, which comes from our sarcoplasmic reticulum because of the arrival of an action potential, and it also needs ATP, which provide the energy. So if we compare side by side the electrical signaling in neurons versus skeletal muscles, they're both excitable. They both have an action potential because they have this ability to move charge across the polarized membrane. Oh, sorry, that got messed up for a second. Okay. What's different is the trigger. The trigger in neurons is something like light, sound, pressure, or neurotransmitter. The trigger in skeletal muscle is always that neurotransmitter, acetylcholine. That local trigger in a neuron is going to open up the voltage-gated sodium channels and cause a depolarization. The trigger in muscles is a little different. So acetylcholine will open up an acetylcholine receptor, which is a cation channel and will allow both sodium and potassium to move. But then the process is the same. So then we open up a voltage-gated sodium channel, sodium rushes in, depolarization. We open up a voltage-gated potassium channel, potassium leaves, repolarization. So the action potential mechanism is the same of sodium and potassium moving. They use different channels. The sodium one in neurons is going to inactivate and have it a refractory period. The one in muscles is going to move in both directions. At the end of the line, the action potential ends in neurons when it reaches the axon terminal. The action potential in muscles ends when acetylcholine is broken down by acetylcholinesterase. Now, at the axon terminal, in neurons, we're going to open voltage-gated calcium channels. Calcium will cause the release of a neurotransmitter. Calcium is important in muscles too. In muscles, calcium released from the sarcoplasmic reticulum is what initiates and opens up action for that contraction. Okay. I hope that was at least a little helpful. Again, if you have questions, please put them in the chat. Okay, I'm going to breeze through a few other topics from earlier chapters. So, active versus passive transport. Active versus passive transport depend on the membrane properties. So keep in mind the plasma membrane, lots of hydrophobic tails. The majority of the space of the membrane is hydrophobic. So small nonpolar things like oxygen gas, steroid hormones, they can just wiggle through the plasma membrane. Things like water that are polar, a little bit can get through, but the majority of water needs help getting through the plasma membrane. And things that are larger, polar, all ions, they really can't get through the plasma membrane at all. And so they're going to need help. And that help comes in the form of transporters and channels. Keep in mind, molecules like to move from areas of high concentration to low concentration. And that we call diffusion. So things are going to move from a very concentrated area to a less concentrated area until they're equal. For salt concentrations, water, it's easier to move water than it is to move salt. And so water will move to areas that are very salty in an attempt to kind of equalize and lessen that saltiness. Okay, so filtration. So transporters and channels. Transporters move slowly because they involve the binding of a solute to a protein. And the protein has got to bind that solute and then change its conformation and allow that solute to move. Whereas a channel is just kind of like opening a door. So where a transporter is where if you've ever taken the subway, you got to swipe your card, you got to move through a turnstile, right? There's a whole process involved. A channel is like opening just a large double door and everyone can just kind of run in much faster. If we talk about diffusion, diffusion again means going from high concentration to low concentration. We have multiple kinds of diffusion. And another way to think about diffusion is passive transport processes. Diffusion doesn't require energy. It doesn't require energy to move from high to low. So we can have simple diffusion, where fat-soluble things, where gases just float on through the plasma membrane, moving from high to low. We can have carrier-mediated or transporter-mediated facilitated diffusion, where things bind in a transporter and move from concentrations of high to low. We can have channel-mediated facilitated diffusion, where things move through a channel from high to low. And then we can have osmosis, which is a special form of channel-mediated facilitated diffusion, where water moves through an aquaporin, again, down its concentration gradient. Again, diffusion processes, those are passive transports. So simple diffusion, channel-mediated, transporter-mediated, things are moving down their concentration gradient, so they're passive. Whereas if you want to move something against its concentration gradient, one, you need to have a transporter, and two, you're going to need some energy. So we call that active transport. The transporters that do active transport get a special name called pumps. And so pumps actively transport against a gradient. They use that binding mechanism. Most of them use ATP as their energy source. Remember, ATP made in the mitochondria, powerhouse and cell. Some of them can use light, a lot of times in plants. And others are called coupled pumps, where they use the energy provided by one molecule moving down its concentration gradient to move another molecule against its concentration gradient. And in anatomy, these are really important in places like the kidney, where you got to move a lot of salts around, but we don't really cover coupled pumps in this class. Okay, move to a new topic, structure of the nervous system. Keep in mind, structure of the nervous system. We have our central nervous system, our brain and our spinal cord, and then we have our peripheral nervous system, which are all of our cranial and spinal nerves, which branch out and innervate our body. So brain and spinal cord, nerves. Brain and spinal cord, nerves. Break the nerves down more. We can talk about the afferent sensory nerves. These are nerves that send information from your body to your central nervous system. So they approach the central nervous system. And then we have nerves that leave the central nervous system with their tasks. So you can think sensory nerves are saying, this is what's happening in the body. Motor nerves are saying, this is what we're going to do about it. They're the efferent nerves. They leave the central nervous system. And those can be broken down into voluntary versus involuntary processes. So somatic system our nerves that are going to innervate, are going to turn on our skeletal muscles, and these are voluntary nerves. And our involuntary pathways turn on involuntary muscles, so cardiac and smooth muscles, as well as our glands. And that involuntary system can be further divided into what the purpose is. So in our sympathetic system, the purpose is to gear yourself up for a stressful situation, a fight or flight response. Whereas in a parasympathetic, the purpose here is to bring you back down to homeostasis, to rest and digest. Okay, I said all of this. Structure of the year. Next topic. I know we're switching gears fast here. I want to get through all these questions in a timely manner, and then if you're watching the video, you can then pause it at your leisure. So let's talk about the structure of the year. We have three major parts: the external, the middle, and the internal. The external and the middle are just involved with hearing. So if we follow the path of sound here, we're going to start with our auricle, which is going to funnel the sound into our external acoustic meatus, which is then going to take the sound, it's going to hit our tympanic membrane, it will vibrate. We will then use our middle ear, which is an air-filled compartment, to strengthen that sound wave so that it's strong enough when it enters the fluid of our inner ear, it doesn't dissipate. So we're going to use our ossicles, our malleus, incus, and stapes, to strengthen that wave function and pass it along to the oval window, which marks the opening of our inner ear. To equalize the pressure, we also have our pharyngotympanic tube, which helps equalize the pressure in that air-locked middle ear by connecting it to the throat, and it opens when you do things like yawn or open your mouth really wide or pop your ears, or equalizing that pressure. Inner ear is where we have both hearing and equilibrium. So for hearing, the fluid in the oval window is going to move the tectoral membrane in our cochlea, which is going to bend hair cells, and it will bend short hair cells near the oval window if it's high-pitched, and it will bend long hair cells away from the oval window if it's low-pitched. The duration of the hair bending will give your brain some information about volume. And all of that will be sent on our vestibulocochlear nerve to our brain for temporal lobe to be processed. For equilibrium, we're talking about things moving in our semicircular canals and our vestibule. So the semicircular canals are these things that look like little horseshoes. They're in the X, Y, and Z plane. And in dynamic equilibrium, or rotations, or moving around, the fluid will move in the semicircular canal and will then bend hairs to tell your body she's moving this way, she's moving that way. The vestibule is where we have static equilibrium, and static equilibrium is your head in relation to gravity, so the up-down position. And in static equilibrium, we actually move little salt crystals, little otoliths that will bend a hair and will tell your body she's bending her head down. Okay. Let's briefly talk about epithelial tissue. So epithelial tissue is the easiest to classify because it's just based on what it looks like. So simple epithelium, one layer. Stratified epithelium, lots of layers. And then you give it a shape. So squamous are flat cells, cuboidal and columnar, their name tells you what their shape is. Simple stratified, squamous, cuboidal, columnar. Okay, and their structure gives them their function. So squamous cells are very thin, so if you just have one simple layer of squamous cells, for example, lining your alveoli in your lungs, it allows for fast diffusion. For your lungs, you can quickly take in oxygen. That's the goal. If you have lots of layers of squamous cells, well, then these are quick and easy to discard cells. They're not energy demanding. You got a lot of them, so you can put them in areas where it's okay to lose cells like your skin, friction points in your esophagus, etc. Cuboidal and columnar cells, on the other hand, are larger and they have more volume to make stuff. So this is where we're going to secrete things, we're going to absorb big things. These cells can also be ciliated, and the cilia will propel stuff. And then the last category is transitional, where we change our shape depending on the volume. Okay, let's briefly talk about some membranes. Cutaneous membranes, these are dry, exposed to air membranes. This is your skin. Those are what we spent most of the integumentary system talking about, was our cutaneous membranes. You got mucous membranes that line open cavities. These are wet. They're things that are open to the air, so like your lungs, your mouth, etc. And then you've got serous membranes, which line our closed cavities. So your internal organs are lined with the serous membranes, and they have two layers, a visceral and a parietal layer, and they're separated by fluid to make them extra protective. Okay. Synovial membranes are the last kind, and they line our synovial joints. Again, the fluid helps create that smooth surface to allow for movement. Okay. So I want to know about glands. So we've got two different major types of glands that exist, but only one type we covered in class. We've got endocrine versus exocrine glands, and they differ depending on where the substance that they made is released. So endocrine glands release their substance, in this case it's a hormone, into the bloodstream. And we didn't cover these because with everything happening, I just cut that chapter because we didn't have enough time. Whereas exocrine glands are going to release the substance onto the surface of stuff, okay, the surface of your mouth, the surface of your skin. And there are many different types of exocrine glands that are classified by how they release that substance and what the substance is. So there are two ways you can classify them. And we focus on this subcategory of exocrine glands called our cutaneous glands. And these are glands that secrete stuff onto your skin. Skin here being the surface. So we talked about sebaceous glands, which are, again, all this are cutaneous glands. Okay, sebaceous glands are our oil glands. Sebum is the oil. It's everywhere except for your thick skin, so not on your palms and your soles. And they empty into a hair follicle. So it's going to release oil onto a hair follicle to lubricate the skin and to kill off some unwanted bacteria. Whereas eccrine glands, these are your sweat glands. They're all over your body. They're going to make this fluid that helps regulate your heat. So when you release fluid, the evaporation of that is cooling. There's also some thought that the pH of it will inhibit certain bacteria from growing. Whereas your apocrine glands, these are sweat glands in your armpits, your axillary region, and your genitals. So these are special smelly sweat glands. And these special smelly sweat glands are larger than eccrine glands, and their secretions are normal sweat plus fatty acids and proteins. And what you're smelling is the bacteria that eat those fatty acids in particular. So you're smelling the bacteria eating your sweat. That's what makes your sweat so smelly in your special smelly sweat regions. Say that 5 times fast. The exact function of apocrine glands is, controversial. Some of it has to do with pheromones and the release of hormones to attract a mate. So your sweaty musk might actually be attracting a partner. But the exact function, again, still unknown. Okay, so much to know about the structure of proteins. Proteins are the things that do the work of the cell. They're made of amino acids. And again, structure relates to function. So we have that primary structure, the amino acid sequence, which is then either twisted into helices or bent into sheets, or secondary structure. Tertiary structure is how that all comes together to form a glob. So we're going to take all of our helices and our sheets and our unformed regions, and we're going to mush them together into a globular protein. And then if that globular protein works with other globular proteins to do something, like they act as an enzyme or they form a really big channel, that is our quaternary structure. So all the subunits coming together. So this is what that looks like. Again, the amino acid has that structure of an amine and an acid with the changing R group that gives it its fun chemistry. And then we have our primary structure, sequence of amino acids, secondary structure, helices and sheets, tertiary structure, all those helices and sheets coming together, quaternary structure, globs coming together. All those globs subunits. Okay. So much to know about condylar versus saddle joints. They're similar, but they differ in the degree of movement. So our saddle joint is our knuckle joint, and we can move it in a lot of directions, but we can't make a full circle. So if you try to make a full circle, it comes out as more of an ellipse. Whereas your saddle joint, because of the shape, it has a greater degree of motion. So you can move your saddle joint, your thumb, in almost a 360 degree, not quite. It's got a greater degree of motion because of the shape of that joint. Okay. Someone wants to know about the cerebrum. So your cerebrum is the outer part of your brain that we divide into 4 lobes. And so if a question asks you, like, what lobe does this occur in, or what lobe is implicated, cerebrum is not the correct answer, because a cerebrum is all the lobes. Okay, so you have to get more specific than just cerebrum. If we look at the cortex in the cerebrum itself, in our 4 lobes, a lot of the function is on the outer edge, the outer surface, the cerebral cortex. And so a few different functions we have in our parietal lobe. This is where we have the touch receptors send information, so we get the feeling of touch. And they'll pass that information to the nearby motor region in your frontal lobe. So your touch comes into your parietal, and then the motor is sent out from nearby in the frontal lobe. We spend a fair amount of time talking about speech. Broca's area in our frontal lobe. This is for word vocalization and how we make speech. So if a person has Broca's aphasia, they understand the conversation, they just can't form the words. Whereas Wernicke's aphasia, this is an issue with comprehension. This is in our temporal lobe, and so they're making words fine, but the speech is nonsensical. They're talking really fast, and none of it makes any sense. The comprehension is gone. The meaning behind our words. Finally, we talked about our anterior association area, which is also sometimes called your prefrontal cortex. This is the very front of your brain. And this is the area of your brain that really separates humans from other mammals. This is where you're able to think through the consequences of your actions. You're able to have working memory, judgment, problem solving. And for here, this is where we have like artistic language comprehension. So a deep, deep meaning to words in the form of poetry and lyrics and novels and the nuances of speech. So the difference between like and love and all of that is contained in our primary frontal cortex. So we're not just making sounds and understanding sounds. Other animals have language. Dolphins speak to each other all the time. They make sounds. That sound tells them about, what is happening and mating behaviors and all of that. But human speech is a lot more complicated and it has a lot more nuance to it. And that's because of our anterior association area, because of the very front part of our frontal cortex. Okay. A few other functions. Vision, again, is in the back and then occipital lobe. And then the temporal lobe on the sides, we have hearing and olfaction. Okay, so that is it for the review session. Stop recording.